slightly different names. Okay. And probably the most common type that you'll see in mammals is this one here called N-acetyl neurominic acid, or Nu5AC. One of the key things for this talk is that it has a negative charge carboxyl here. This is a glycerol, which is added at one side of the molecule, and it has an amine as well. So it's an amine sugar that has a charge. And so the cell surfaces are projecting that negative charge. All right, now nothing seems to want to work. Okay. Okay. So why is that useful for bacteria? Well, if you have a bacterial infection, particularly if it's a bacterial infection in a mammal, then it turns out that those bacteria have evolved the ability to scavenge sialic acids from the cell surface. And why do they do that? Well, they secrete enzymes that go off into the solution, and then they cleave off the sialic acid from the glycans, and then they import the sialic acid into the cell through a transporter. And then when it's in the cell, it can do multiple things. So firstly, because it's a sugar, it can be used in glycolysis and to generate ATP and energy. Some of the intermediates within the pathway can be used to make peptidoglycan, which is important. Some crafty bacteria actually, including the one we're going to talk about today, Haemophilus influenza, have developed the ability to place those sialic acids at the terminal end of their own sugars, and they do that to mimic in biomimicry, and they do that in order to get around the innate immune system. So the innate immune system will see these bacteria and recognize them as self. So it's a way of evading the immune response. And then if you are a bacteria that makes a capsule, some of the intermediates are also involved in capsule formation. So sialic acid is a really important molecule for bacteria during infection. It's not critical for growing the bacteria, but it certainly is important for colonization and for pathogenesis. So one of the examples of this is Streptococcus pneumoniae. And in this experiment here, this is a paper that from a few years ago, they took mice and they were taking different types of amino sugars, so either glucose, glucnac, that's an intermediate, galacnac, sialic acid. This is a glycol, this is a variant of sialic acid, or sucrose. And if you add a little bit of these molecules into the nasal passages of these rats, then you can see that only with sialic acid do you get a really strong pneumococcal response to that. So that means that sialic acid is able to colonize at least the mucosal surface. If they then looked at the lungs, what they found was only when sialic acid was present or added to the rats that you found a significant amount of Streptococcus pneumoniae in the lungs. And not only that, the sialic acid was promoting biofilm formation where the other amino sugars weren't. So that hints that actually sialic acids are probably important for pathogens. And these types of experiments have been done with other types of pathogens as well. So that's what got us going to explore sialic acid. And most of our work had really focused on the enzymology. James and Rachel both published papers down here, and Michael, in fact. That was really fun. But I guess one of the things, the nagging parts of the pathway, is that we didn't really know how sialic acid was being imported into the cell. So this is a gram-negative bacteria. Just going into a bit more detail. Here are your host conjugates. This would be the outside of the cell. This would be a sialic acid. Sialidases are exported by the bacteria to cleave them off. If you're gram-negative, you go through a passive transporter, an OMP transporter. Some bacteria have their own sialic acid passive transporters in the outer membrane. That goes into the periplasm, and then it goes into the cell. And so we had worked a lot on many of these systems, including the regulation of this pathway. But really the nagging question was around how this transport worked. And so I guess back in 2017, we had worked on a sodium solute symporter. And that's because in bacteria, they use different types of transporters. So some bacteria will use a a major facilitator family transporter. These use a proton gradient to drive sialic acid import. The sodium solute symporters, of course, use a sodium gradient in order to drive symport. This was our introduction into transport. There are also ABC transporters. These are a little bit different because these are primary transporters because they use ATP energy, so the hydrolysis of ATP, to drive the transport mechanism. But they also have a substrate binding protein that captures the substrate, in this case sialic acid, with high affinity, and transports it to the membrane to be transported through the membrane by these other domains here. And so trap transporters, actually, if you look across these three ATP binding cassette transporters, these were generally well known. They're found in many domains of life. Sodium solute transporters, again, the mechanism by which they work is generally known, and the major facilitator superfamily, again, were generally well known. There are lots of examples. But the trap transporters, they were structurally unknown. We'd known about them from sequence studies even back in the mid-90s, 1990s. And so they were kind of an odd combination. So they're a secondary transporter in that they use a sodium gradient, but like an ABC transporter, they also need a substrate binding protein to connect the substrate to the membrane and then transport it through. These two domains seemed a little bit unusual. Their sequence wasn't similar to any of these other three families, so they seemed unique, and they definitely didn't use ATP. So the question arose about how is it that these these trap transporters function? And I guess a lot of these transporters use the alternating access model generally, and there are a few flavors of that model. When we were working with the sodium solute symporter, this is this one here, this family used this rocker switch model. So you have two domains, one here, and they're moving back and forth in this motion to allow access for the substrate to bind and then the substrate to move into the cell. In a kind of subtle play of this, there's the rocking bundle. Here you have a domain that is a stator domain or a scaffold domain that doesn't move, and you have instead a bundle or a barrier or a domain that's moving against that domain. And then a relatively new type of mechanism that was put forward probably in the last couple of decades is the elevator mechanism. And here again, you have a scaffold or a stator domain, but instead of moving back and forth, here you have a domain that is moving up and down through the membrane. This is the elevator. I'm guessing from the title that you'll know which one it is, but back in when we started this project, we knew that it was a sodium transporter. We knew that the P subunit was important for function. We didn't know anything about the membrane protein domain and how that worked. And so our hypothesis back in 2017 was that it was a rocker switch model. And this was perfectly sensible. We didn't really know what this Q domain in green did, but so the M domain, it could quite possibly be functioning as a rocker switch, but the difference being that you had this substrate binding protein, and this is not dissimilar to how the ABC transporters work in a way. All right, so that's where we started. And to get things off, to get things going, what we did was to try and express a whole number of these trap transporters. So we've got a bunch of different genes. They're not all sialic acid transporters, actually. They're just trap transporters from various bacteria. And perhaps to our surprise, when we try and overexpress these bacteria, these transporters in E. coli, many of them did actually overexpress reasonably well. And we thought that this would be way more challenging. And so the first one I'm going to talk about is this one down here. It was the one that was the best expressed. So that was the one we started with. And this was from a bacterium called Photobacterium profundum. Now, this is not an organism that that we know of, lives in mammals, but it certainly does have sialic acid metabolism, and it does have a trap transporter that imports sialic acid. So it lives in the sea at about 15 degrees and at various depths, so it functions under pressure as well. Now, the one thing I say here is that it's not fused. So if you look at the operons, or the way the proteins are expressed, there's a single transcript that expresses first the Cy-P, then the Cy-Q, and then the Cy-M. In some cases, and we're going to talk about this for the Haemophilus influenza, the Cy-Q and the Cy-M are fused together with a sequence, but this one, or many of them actually don't have that fusion. So we call this a non-fused example. All right. So I guess the first experiment, or the first question that we had, was what is the stoichiometry or the oligomeric state? So there could be 2 Cy-Qs to 2 Cy-Ms, 1 Cy-Q to 1 Cy-M, 3, we don't know. And so the first experiment that we did was a sedimentation velocity experiment. If you've not seen one of these before, these experiments are very simple. We're placing the protein that has been purified. In this case, it's in LMNG detergent. It's actually quite stable. And we're putting it in a centrifuge, and we're spinning it at about 50,000 RPM, so quite fast. And of course, things that are really big, large proteins, will sediment or move through solution quite quickly. Things that are small, like these guys up here, they don't move quite so fast. And so when you do this for a purified Photobacterium profundum, you get a nice data profile that looks like this. This tells us, when I look at this, I see that there's probably one, maybe a few minor species that are moving through solution or sedimenting. And when you fit that to a model, what you see is that if you measure by absorbance, you have this blue line, and you come out at about a size of about 7S. Now, one of the problems with membrane proteins, when you do this for a soluble protein, it's much easier because you know all the parameters of the experiment. But when you have a membrane protein, of course, you don't know how many detergent molecules are bound, or actually whether there are any lipids bound. And so it's hard to turn this into a mass. So we say that the size, the sedimentation coefficient, is roughly about 7S. But in this experiment here, we did something that is kind of clever that other people had developed before. And here we're detecting, we are monitoring sedimentation using absorbance. So that looks at the protein. And we're monitoring using interference, which monitors everything that's in solution. So even the molecules, the micelles, that are solubilizing the membrane protein can be seen by this technique. So when you do that, you'll see that in the interference optics in brown, you have this peak here. This is completely consistent with a micelle of LMNG that has no protein in it. And then here you have a peak, which is both protein and a little bit of detergent. And so with some clever maths, we can determine that almost certainly this is a monomer with a stoichiometry of 1 to 1. There's roughly 100 detergent molecules that are bound. So this was really our first structural information, and so this was quite exciting. Right, so at this point, we wanted to know whether our protein was functional. The way we did this was, well, to connect to a really good Italian group, Cesare and Maria Francesca. And they had been working on functional studies for a long time of membrane transporters. And so we sent them some protein. And the first experiment we did, so we have the protein here. It's in a proteoliposome. We use valeromycin to form an electrogenic gradient. So the potassiums that are inside go out. And then we have sodium on the outside, and we have tritiated nu-5AC, so that's sialic acid, and so we can track it. And so if it's functional, the nu-5AC will transfer into the proteoliposome, and we can track that. And so Maria Francesca did all of these experiments. And so if you have if there's no Cy-P present or no sodium gradient, you don't get any activity. And that's completely consistent with what we knew, that it needs sodium and it needs the Cy-P protein to collect the sialic acid and take it to the membrane. If you have a sodium gradient but not an electrochemical gradient, then it's reasonably active. But if you have both, it's very active. And so that seems consistent with what we knew. If you now titrate the amount of Cy-P, we get an increasing rate relative to the concentration of Cy-P. And that tells us that probably the KD for Cy-P to Cy-QM is probably in the somewhere in the micromolar range, and I'll come back to that a little bit later on. So our protein was functional. That was great. It's a sodium transporter, so it's taking these sodium molecules. So we're curious to know how many sodium ions were being transported. And principally, this was because this is a negatively charged molecule. So we thought that at least 2 molecules of sodium must be transported for it to be electrogenic. And so here, what Maria Francesca had done is she's titrating concentration of sialic acid against activity, and you get this nice sigmoidal curve. And that suggests to us that there may be multiple sodiums being transported per sialic acid. But just to verify that, she did another experiment where inside of the proteoliposome there is a dye that fluoresces in the presence of sodium. And so now you can directly calculate the amount of sodium ions that are transported per sialic acid, and it's about 2.5. So it's a sodium transporter, it needs Sci-P, it's functional, and we have a good sense of the number of sodiums. All right, so I guess during all of this, one of the things that James and Michael and Rachel to an extent were doing, were trying to crystallize these trap transporters. And surprisingly, they did actually form crystals reasonably readily. However, none of them diffracted. And so the way forward here was to try and solve the structures using Cryo-EM. But of course, and probably this community knows this, one of the problems in Cryo-EM for membrane proteins is that when you have it in a micelle, for example, detergent or amphipol, often for these transporters, there are some examples where this is not the case, but there's not that much outside of the membrane. And so for orienting the particles, this is a bit of a problem. And so the way we got around that is that Pete Mace and his brilliant technician, Sam, they just developed a mechanism, well, they had adapted a mechanism from other people to generate these nanobodies down in Otago. And so we sent some protein, and this worked first time. So the protein was fluorescently labeled. They panned a bunch of nanobodies, and they found some. These were yeast. Are they yeast? Yeah, I think they're expressed on the surfaces of yeast. And so we got the sequences, and we expressed the nanobody. When I say we, I mean James and Michael in this case. And actually, we also added this hop Q to make it a little bit bigger. So that makes the whole particle quite large. And now in the Cryo-EM, we could orient the particles. And this was an example of the types of particles that we saw. So these are quite encouraging, and you can see quite a lot of detail within these images. Just to say that we also looked at what the KD was for these nanobodies. We have about 15 that seem to bind. We've only looked at 4 or 5 of them, but they're all roughly in the low nanomolar range, 10 to 100 nanomolar KD affinity. So they bind quite tightly, and we could show that. All right, so now that we had the particles, I guess there'll be a few folk here that know a thing or two about Cryo-EM. And so we had a bunch of different classes. You can see actually that you have quite a lot of mobility, for example, in this class here for the nanobody and the megabody with the hop Q. And it turns out that you don't see the hop Q, but you do see the nanobody. Roughly half of the particles looked like they might have a nanobody attached. And then going through the processing, we end up with roughly a 3 angstrom structure. And that was really exciting. To see the local resolution in the inside is down to 2.2 angstroms resolution. We could model virtually all of the trap. We could see almost all of the side chains, and we could see some other features that I'll talk about in a second. That gave us a good sense of how these traps work. So this was the structure. In this case, we used amphipol, and our thinking here was that amphipol would make the structure nice and steady, and so you'd have good particles. And it turns out that the reviewers of our paper thought that we should also do it in a native environment. So we've also solved the structure in a God, I've forgotten the word now. What is that thing? Yes, in one of these things here. In a nanodisk, gosh. Right, so let's just orient the structure. Here's our megabody. We can only see the nanobody part. This is the M subunit, which I was showing before. So that was one of the genes. Here's the Q subunit in green. And one of the first things that we saw was that the Q domain, which forms the scaffold, was a unique structure for a membrane protein, and it sits on this angle. And then the scaffold part of the protein is formed by parts of the M subunit, so the scaffold 1 and scaffold 2. And then we had what looked to be a transport domain, which is made up of 2 parts, so transport 1 and transport 2. And so when we had this information, suddenly we could see that this wasn't, our hypothesis was that it was a rock switch, but actually that was probably not right. And only an elevator really makes some sense. We've got a transporter domain that looks a lot like, in structure, the DAS dicarboxylate transporters, and they use a transporter domain. So almost certainly we think these use a transporter domain. And so here's the transporter domain in the middle. Unlike the DAS family, it's not a dimer, it's a monomer. So that was a little bit unusual. And again, we'll come back to that a little bit later. So with the transporter domain, we could look to see what other features were there. And so in the DAS family, you have 2 binding sites for sodium that sit either side of a pocket, which is the substrate binding site. And similarly, for our trap transporters, the transporter domain has the same topology. And in fact, we could see cryodensity for each of the sodiums on either side, which was really encouraging and great to see. One of the things we could do, even though most of the connections that the sodium's making to the protein is with main chain interactions, there are a few side chains, so there are a few mutations that we could make. And if you make those mutations, Maria Francesca and Cesare showed that activity was blocked. So here activity is about 3 or 4%. So if you make mutations on either side, then it's blocked. So that adds to the idea that these are real sodium binding sites. We also made a couple of mutations in the substrate binding site. This is actually a double mutation here. And again, you lose activity. So we think that's perfectly sensible for where the substrate binds. So one of the other experiments that we did, so just coming back to the point that we had also solved the structure in a nanodisk. And so the nanodisk, of course, has phospholipids embedded, so it mimics the native environment. And when we looked at this structure, one of the things we could see is a large number of cryodensities that are probably most consistent with lipids, phospholipids binding. Now, one of the experiments that Maria Francesca and Cesare did was to compare the activity in proteoliposomes that have egg lipids versus E. coli lipids. And they showed that the E. coli lipids, when they're present, you get much more activity. And of course, when we're looking at this density, these really do look like lipids. Now, the best described lipids in terms of the density sit on the scaffold domain, and they intercalate these aromatic residues at the top. And so we think that these residues are here because the state of the scaffold domain is set in the membrane. And so that's holding the membrane, or this part of the transporter, within the membrane. But we do see some density that would be consistent with maybe greasing the movement of the transporter domain. And so you see at some places where you have the scaffold over here, the transporter domain here, you can see density for lipids on that side as well. So maybe they're also helping with membrane movement of the protein through the membrane. Around this time, Jane Allison got excited about this work, and she wanted to model some of these motions. So this is a MD simulation. It's an all-atom simulation. And so we have a membrane that I'm showing here. I'm not showing the waters. We have the transport domain that's moving up and down, and we have the scaffold domain that's set in the membrane. And one of the things you notice is when the membrane is being pulled up, this is not an MD simulation, this has been pulled in that motion gently, you see that there's quite a large change in or deformation of the membrane around that transport. And so I'm guessing that there's some kind of energetic cost for doing that, or perhaps the MD simulation is not quite right, but it doesn't quite fit with what's going on with the elevator here in terms of energy costs for moving up and down. So there must be some mechanism to try and grease those motions, or there's something interesting going on around those membranes during that motion. All right, so I'm going to change tack a little bit here. And really, this is the work that Michael led. And so he was interested in this one over here. And this is a little bit different in that it's a fused tract transporter. And so again, you have your transcript, you transcribe and translate your Sci-P, and then your QM are connected by a sequence of residues. And unusually you get, just for these fused variety, you tend to see on an SDS page gel, this is actually a Western, you tend to see two different bands, and none of the other tract transporters seem to do that. So if it's a non-fused, you don't seem to see that, at least in our hands. And so here is an SDS page here, again showing HI Sci-QM. So this is Haemophilus influenza, sialic acid transporter. We see two. If he does a Western, we see these two bands here. And at least what could be happening here is you could have cohabitation. So it seems that what's going on is that it's really dependent on the amount of detergent that's in the micelle. So if you have a high concentration of LMNG, which is the detergent we've been using, then you get a peak at around about, I don't know, 65, and you have a shoulder to that peak. But if you drop the LMNG concentration, we're still above the CMC level. Now we're driving it to what seems to be a larger sized particle. And so I guess that's consistent with seeing two peaks here in the SDS. Just to say that for the non-fused, this is the PP Sci-QM that I showed you before, when we have it in the same concentration of LMNG, we don't see this drive to a dimer formation in the SEC trace. So it seems to be something that's unique to at least this fused tract transporter. Now, Michael went along and just to verify that this was a dimer, it could have been a trimer as well. And so he did an analytical centrifugation, sedimentation velocity type experiment looking at this peak, but actually I'm showing you here, the second peak down here, looking at the difference between the interference in green and absorbances in pink. And when you do this calculation, you see that it's only consistent with a dimer. Trimer gives you a very asymmetric molecule that is probably not likely. And so a dimer is the most likely explanation for that. And so probably the two peaks that we're seeing are a monomer and a dimer. And the reason that's useful is that now we might be able to solve the structure of a tract transporter without the need of having a nanobody there. And the reason we wanted to do that is we were worried that the nanobody was trapping the protein in one particular conformation. And we've just saw in the EMD simulations that the protein can move between two different conformations. So Michael was able to solve the Cryo-EM structure. And actually we get two different conformations that are about 50-50 in the particles. One that we would think is biologically relevant, that's this one here, and this one here that we're in the micelle, it's flipped 180 degrees. So we can see both of those, so the parallel and the anti-parallel. And this is more akin to the DAS family transporters that use the elevator motion. And here, though, they have a much more intimate connection. What we see instead is a lighter connection where there's clearly lipids at this interface. There are some connections between them, but it's certainly not an extensive interaction. Why might that be useful? Oh, just to say that the structure of the fused is largely the same, except it has this extra fusion helix between the two that we could see. That extra fusion helix actually is also at that interface, but otherwise it's largely the same as the non-fused PP example that I showed first. So this is some data from Simon Schuring's lab. So it's a high-speed atomic force microscopy. And so in a membrane on the instrument, you can see the tract transporters do in fact interact with one another. And so why that might be useful, I think, is maybe like the DAS transporters, the idea is that if you have the stator or the scaffold domain set and bound to two, then it can move more easily up and down without any motion in the stator. So it's more stable for that transporter motion. Why the PP doesn't do this, we don't know. All right, so coming towards the end, we're interested actually in the full complex, and I've only talked about the Q and M domains, which are these two here. What about the full complex? How does that work? Well, first thing we wanted to do, of course, was to take our particles, soak in a high concentration of P, and hopefully we'd be able to get the full complex by Cryo-EM. But it turns out that, at least by all of the biophysical methods that we've tried, the affinity for the P, for this component to the membrane components, is very weak. So we measure it roughly in the mid-micromolar range. And so that makes it really tricky to do cryo-EM, because you'd have to have an enormous concentration of Psi-P present to see any particles. And we did that for both the HI and the Q. And that actually fits, going back to our functional data, that fits with the fact that as you increase the P concentration in the activity assays, you're still getting increasing activity. When we did that for the HI Psi-QM, we see two peaks, because I've told you that it's a dimer. So we see a monomer peak, which is binding 1, or a dimer peak that's binding 1 or 2. We can't tell. And again, when we measure the affinity, it's in the mid micromolar range. I haven't talked at all about the Psi-Ps, and because we're going to talk about the complex now, I thought I'd just mention that these proteins are highly stable. They diffract really well to roughly 1 angstrom, and they go from an open conformation, and then when the sialic acid binds to a closed conformation, so like Pac-Man, if you've played the game, and that makes lots of contacts with the sialic acid in that cleft up in here. So when sialic acid's bound and it closes around, there's a lot of connections here. But just want to make a point here that this is the closed conformation with sialic acid. Here's one domain on the right, one domain on the left, and there's still a crevice that's full of water molecules, and these are highly ordered and also quite conserved across P constructs. So because these are so well described, there are a lot of structures of these P subunits out there. And so these water molecules are a feature, actually, of these molecules. And I'll come back to why that's interesting in a second. So what Michael, this was pre-AlphaFold, but what Michael and James had done was to map what this might look like. AlphaFold actually has much the same answer. And so we have these hotspots on both the P and the QM. We could make mutations on either the QM or the P. And in most cases, most of these mutations ended up losing activity. So that suggests that this complex is probably about right. And so if we're able to place the P on the QM, so over here, we've got our structure with our P structure. Here we've got the transport domain is in the down conformation, and so the substrate binding pocket is facing into the cell. The P subunit is docked onto the surface. This would be the periplasm. And so what we think happens is that the transport domain, as it moves up, now the substrate binding pocket is here. So that's perfectly placed to accept the sialic acid from the P subunit. And these motions, we don't have the structure of the up conformation, so we modeled these motions on one of the structures for a DAS family complex that is in the up conformation. So the transport has moved up. So that's our guess of how it works. And so this was a morph model that, in the way that we think this happens. And so the transport domain goes up. The substrate binding protein has to go from a closed conformation to open conformation, so the substrate can go into the substrate binding site. Like I said, this is one of the questions that we have on how this functions. So in ABC transporters, at least, the same question arises. How is it that the substrate and the substrate binding protein binds generally with reasonable affinity? How can it move into the transporter? And in ABC transporters, or at least in this example, what happens is you have this loop here, which they call a scoop loop, which binds the substrate and draws the substrate down into the transporter pocket to be transported once you have ATP hydrolysis. Well, in our structure, we don't have that same kind of loop. And also, it doesn't reach right up into, we do have motions that change, but they certainly can't reach the substrate in the P subunit. And so what we think, this is actually another transporter that we've been working on, so this is unpublished data. But what Michael has shown is that you do have a loop when you're in the up transporter that pokes into that crevice where all those water molecules are. Now, we know those water molecules are really important for the affinity to sialic acid. So what we think, or our hypothesis is that, or this residue, or a residue like it, disrupts those water molecules. So you go from the closed conformation to the open conformation. And now with that water molecule network disrupted, the dissociation constant for the open and closed is changed, and now the sialic acid can move into solution. Move into the pocket. And that's just to remind you that the water molecules that are at that crevice. All right, so let's wrap up. Started off talking about how transporters work, and we're focused in on the trap transporters. And so what we think happens is, to use an analogy of an operator and an elevator, is that the operator, in this case, the Sci-P, binds the passenger, in this case, sialic acid, takes it to the elevator. The operator pushes the button for the elevator to move to position. The door to the elevator opens. The operator is ready to load its passenger, in this case, sialic acid, into the transporter. And then the door closes and the elevator is sent down to the bottom floor. In this case, the elevator domain is moving down and delivering the substrate into the cell. And so that's where we're at, I guess, but there's still a lot of open questions. What is the order of events? We don't really know when sodium binds. We don't know when the substrate binding protein opens. We don't know when the up conformation occurs. We don't know what the up conformation looks like. So when the elevator's in the up conformation, what does that look like? We don't know what the complex looks like. And how does it open Sci-P, although we have a hypothesis to test. How is sodium coupled to these structural changes? We know where the sodiums bind, but it's less clear on how they actually deal with motion. And of course, we've got this interesting feature that we have these lipids bound. And so what they're doing is also of interest to us, and how that's important for the transport mechanism. And so that's where we are now. And thanks, that's the end.
Thank you for the great and interesting talk. Are there any questions from the audience? You can also just unmute you and ask them directly, or write them in the chat. So right now I don't see any, so I would ask a question. You said that those transporters are not existing in mammalians. So
No.
would it be possible to use now those structural data to create some sort of new antibiotic or something similar?
Yeah, that's exactly right. And so we're doing that. So we have we're focusing on the Sci-P, so the transporter protein. The substrate binding protein, and we've been using screens and to try and find molecules that bind. We also work with a group that in Sweden that carbohydrate chemists, and they've been making sialic acid analogs, of which there are many, of course.
Okay, cool, interesting. Yeah, Patrick.
Just a quick question. Back when you were doing the structure with the megabody, where you really didn't resolve the hop Q domain, did you also look at just the nanobody alone and whether or not you could do the structure without the hop Q fusion?
Yeah, not for that one, but we have subsequently. We started looking at some of the other nanobodies, and in the hope that they might capture other conformations, bind either at a different surface. And in those cases, we didn't use the nanobody. They bind perfectly well, and you can solve the structures happily.
Yeah, thank you.
Are there any more questions? Otherwise, I have another one. Like you said, a lot of bacteria get the sialic acid from their host, like from the mammalians. Is it known from where the Photobacterium profundum gets it? If it's in the deep sea?
That's a strange one. We don't know why. Maybe it has a mammalian host that we're unaware of.
Oh, okay.
That's my guess.
Cool. Yes, Cindy?
Hi, it's a very interesting talk. Thank you very much. So I'm quite interested in the concentration of LMNG. So you said that high LMNG concentration will improves the polymers. formation, while the low LMNG will favors for the monomers. So is that a common symptoms for all the membrane proteins, or is like just for this transporter?
Yeah. So I think it's the other way. So if you have a low concentration of LMNG, then you tend to have an oligomer. And if you have a high concentration, then you tend to have a monomer. And I think the reason for that is that you have more LMNG molecules around. So you need roughly 100 molecules in each micelle, roughly, for the monomers.
Okay.
That's about how many we find. And so if you drop it down, you get it. So say if your concentration relative to the protein is say 80 molecules per protein, then you can imagine that the micelles aren't going to be quite so stable, but they can at least try and solubilize themselves by forming an oligomer. And we call that cohabitation. And that happens, I think, quite readily with most membrane proteins as you remove the detergents, because they need to solubilize somehow. But in this case, we're getting a particle that, or a conformation that is regular. So it's not just it's not a particle that is, I'd say, aggregated, for example.
Yeah, cool. Thank you so much.
Maybe another question from me. So do you think, or do you plan to also use the nanodisk on the transporter from the Haemophilus influenza, or did you already do that?
I actually can't remember if we did that. You need a bigger one. Geez, James and Rachel, if they're still there, might be able to remind me. I don't think we did.
Yes. How?
Hello, can you hear me?
Yes.
Yeah.
Yes, sorry. Wonderful talk, Renwick. With respect to I'm a bit curious on the P subunit, the M subunit. So what's the energy that drives it to, the elevator membrane to go over the membrane to the solvent environment and then comes backward. So, I mean, this is very interesting phenomenon to me. So what do you think about what's the energy to drive these conformational changes?
Yeah, that's a really good question, and one that we don't really know the answer. We know that it's sodium. If you have a sodium concentration and an electrogenic gradient, then it's functional. And so it must be sodium binding to the protein. And we know where those sodiums bind, but it's not so clear what the conformational changes are between the up and the down. Until we have an up conformational structure or a complex structure, I think it's going to be hard to answer how sodium is driving transport or the protein motions through the membrane.
Yeah, thank you.
Is it possible to cross-link the soluble fraction to the membrane fraction to create a complex, or?
Yeah, it's possible, because the biggest issue, of course, is that the KD is very, very low. That is it seems to be low affinity, at least in our hands. So that's still going to be an issue, but that's a good idea.
I don't see any more questions, and I ran out of them as well. So if there are no more questions, I would say oh wait, Patrick, is it?
Yeah, so just thinking about the process that you need to do with the binding of your soluble protein. Obviously, it's low affinity because the whole purpose is to catch and release, right?
Yeah.
Which is not in some ways dissimilar to what GPCRs do, G protein coupled receptors. They catch and release G proteins effectively. So in our space, it's very hard to capture a complex, and so mostly how that's done is an engineering related phenomenon that traps a specific substate that has high affinity of the two reagents. And presumably for you, at least in your model, and obviously you need evidence for that, but your upstate that engages the closed sialic acid bound, if you could trap that before it releases the molecule is likely where you could do that.
Yeah.
Is there any biochemical or biophysical evidence about manipulations that may be able to trap that state?
Well, so the way we'd gone about it was through different nanobodies, hoping to bind at different conformations, at least the up conformation. The cross-linking idea is a good one for trying to trap different conformations for the transporter. Others have looked at manipulating actually the Sci-P. So at least in our hands, the unbound, so the open conformation of sialic acid will still bind to the transporter, but we think the closed conformation binds more tightly. And so maybe trapping the substrate binding protein in the closed conformation might be one way forward, and seeing if we can optimize the KD there. But it is a little bit tricky.
Yeah, sure. All right, sorry, thank you.
And thank you everyone for the questions. And thank you again, Ren Dobson, for the great presentation.
Pleasure. Thank you.
Yeah.
All right. Thanks, team.